that's turned on yet, and we'll get that started. You're welcome. There you go.
Good afternoon. My name is Mariel from Chimera Labs. This question is for Rachel and Mary, either of you. In terms of the analytical comparability that we are looking for the biosimilars, do you have any acceptance criteria already established? Because we always said about similarity, but how similar? Is it 90%, 95%? And also, we have to have at least 10 different methods. And how is assess if there is any of the method that is not so comparable in terms of how heavy is that in when evaluating the biosimilarity?
So the question you're having, I think, is we don't have any set acceptance criteria for the analytical comparative. Okay. So that would be up to you as the biosimilar sponsor to compare those quality or the ranges that you acquire for the U.S. licensed product, reference product, and compare those ranges to your proposed biosimilar product. So depending on the attribute, depending on several different things to consider, it may or may not be okay that they're not completely overlapping. So it just depends on the attribute, the method, and the...
For example, for potency, if they could be 80% similar, would that be something that is okay, or you expect more than 90 for potency, for example?
I think, again, it depends. Okay.
I think then you need to ask yourself, if you gave a patient a reference product with a particular potency and then you gave them a biosimilar product that's got 80% of that potency, what would happen to that patient? And what do you think about whether that product is biosimilar or not?
Yeah. My question is mostly because when we evaluate potency, usually cell-based assays that they have kind of high variability also, then that's what and it's always an issue how to establish the protocols to put those acceptance criteria. That's what I was asking that. Thank you.
All right, and again, if there's any questions in the room, come on up to the mic. Let's take our first online question, please, Ray.
Thank you, Jeff. How is FDA dealing with reference standards for biosimilars? Is there a preference for internal standards or even a comparator available in the market during the execution of the comparability studies?
To answer the last part of that question, typically for the biosimilar products, no, there are not like publicly available reference standards. Depending on the stage of development, you may start out using a reference standard that is the U.S. licensed reference product or the non-U.S. comparator product. But the idea is that eventually, for the control of your product, that you would develop your own in-house reference standard. And that characterization is dependent on you as the sponsor.
All right, next question, Ray.
What information does FDA expect for cell bank characterization and stability?
So I think for a biosimilar product, the expectation for cell bank characterization and stability is the exact same as it would be for a 351A application. We expect that your cell bank is fully characterized with growth kinetics as well as genetic stability. The LIVCA studies, the end of production cell, the testing, all of that is the same as would be required for a 351A application.
Okay, microphone number 1, nice and close for me, please.
Hi, Michelle Doherty from Data Revive. First of all, thank you for the excellent presentations. And I think it was in Mary's presentation, you talked about the need to assess investigational and commercial lots in the analytical similarity assessment. So investigational and commercial lots of the proposed biosimilar. So can I interpret investigational lots to mean both lots that are manufactured by the clinical process and then also lots that were used in clinical studies?
Yes. So ideally, you want to use lots that were manufactured with a process that's representative of what you're going to be selling, or the commercial process. And you also want to know, you also want to include in there the clinical lots. Any other lots that might be earlier development lots, you'll have to justify inclusion or exclusion, I guess, also. But you'll want to include, you could include them if you can justify that they're representative of the commercial process.
Thank you.
Microphone number 2, please.
For the validation of the immunogenicity and neutralization assays, there are some chapters in the USP that also refer to that and FDA guidance. Then which one oversees, as a priority in terms of designing the protocol? Meet all the, because I have noticed some difference in the requirements between them. Then in this case, which one we should follow?
Thank you. I think, yeah, Susan Kirshner would be a good person to answer that question.
So this is Susan Kirshner from FDA. We recommend, of course, that you follow FDA guidance. However, guidance is a recommendation, and you can, if you can scientifically justify your approach, then that's fine. So there's a lot of approaches that are not in either at USP or FDA that we've accepted, and you'll just have to justify them.
Okay, Ray, can we get another online question?
What data does the FDA expect to demonstrate that an assay is adequate to demonstrate stability?
So to demonstrate that a method is stability indicating per se, you would have to provide, so depending what that assay is and what it picks up, you would have to be able to provide evidence that is sensitive enough to pick up those particular degradants or impurities with adequate sensitivity. So sometimes for say, size, exclusion, purity, you would need to potentially use spiked samples or enriched samples for that impurity to validate that that method was capable of picking up the correct impurities under those conditions. You would want to use a wide range of different samples as well, drug substance, drug product, to be able to capture the full accuracy, precision, and everything of that assay. But if you're going for stability indicating properties, then you need to show sensitivity for the impurity that you're picking up.
Okay, get another one back there, Ray.
Should all functional activity of a product be evaluated for the purpose of determining highly similar?
So you need to understand the critical parts of the product that contribute to its biological activity. And for both the comparative assessment and control of the product, you should be able to test all the critical aspects that contribute to biological function.
But I think to maybe to expand on what Mary's talking about, so in the sense there are potentially some products, maybe a monoclonal antibody per se, where there's an FC portion. However, the intended mechanism of action of that product does not have effector function, is not dependent on effector function. In terms of establishing biosimilarity, we would not necessarily require that you would fully test that and with full with several lots or anything, but if it would definitely need to be part of the characterization. So it's not enough to say, well, the reference product does not have this function, therefore my product doesn't have this function. You would have to show us data for that. So you would have to probably do some characterization studies for effector function in that example to show that your the proposed biosimilar is equally not as good at effector function as the reference product. So, but that would be more of a characterization study and as part of the package.
Okay, another online question for us, please.
What should we expect during inspection of a biosimilar product?
So I think Rachel kind of touched on this a little bit. So you'll have the regular GMP part of the inspection, and then you'll also have like a biosimilar portion of the inspection, and that will be usually at a site where you perform the assays or the testing that contributed to the comparative analytical assessment part. And what we'll be looking for there is data traceability and whether the data are accurate and if the things we see at the test site are the same as the things that were submitted to the BLA. And wherever the biosimilar inspection ends up being, you'll be expected to be able to provide all the data that contribute to the comparative assessment. So let's say we went to a site where only MS was performed, you would still be expected to be able to provide all the rest of the data, the potency, purity, everything else, and then also be able to answer questions about all the rest of the data also.
Okay, another one, please, Ray.
What is the FDA perspective on mutation impurities and their presence in biosimilar products?
I'm sorry, can you repeat that? I'm sorry.
Yeah, the first part again. Well, the whole thing again, please, Ray.
What is the FDA perspective on mutation impurities and their presence in biosimilar products?
So, yeah, excuse me, I mean, like maybe sequence variants? Okay, so I think, and then Susan can chime in, I think for any type of impurity, the idea would be that if you can detect it, you test it, you would have to characterize it, and depending on the impact of that variant on potency or other aspects of the product, you may have to control it. So I don't know that we have there would be a hard decision made about that, but you would have to adequately characterize that impurity, show that it doesn't impact potency, and be able to control it if needed.
So if you're talking about mutation variants that are expressed, there is an expectation that you would have the same sequence, same primary sequence, with minor modifications like at the N and C terminus are allowed. So if you have sequence differences, amino acid sequence differences between the biosimilar and the innovator, that could potentially be a problem if they are not also present in the innovator product. And then it becomes a quantitative issue, not a qualitative issue. If you're talking about sequence that, variations at the level of the gene, there is no expectation that you'll have the exact same genetic sequence for the biosimilar and the innovator product, because there is redundancy in the genetic code, and that's allowed.
All right. We have another question over here at mic 2. Thank you.
My question is in regards to biosimilars that are already approved in Europe. To be able to approve them in US, would you be able to use the same analytical data that was already presented in the filing in Europe and then just do a minimum clinical development, clinical trials in US?
So you'll need to have a U.S. license reference product included in your assessments. But I like just reusing, like outside of that, I don't know if there are any.
I mean, yeah, you would have to test the EU product, the US product, and the proposed biosimilar in the three-way analytical bridge. So you would have to show similarity between the proposed biosimilar and the US reference product, the proposed biosimilar and the EU product, and the US and EU product. Is that what? Okay. Do we understand the question?
Yes.
You said you're saying a biosimilar product that's approved in the EU, presumably against an EU comparator.
It could be either.
Oh, I see.
Because there are the two options against the UE competitor, but if, for example, it's a biosimilar that is against the United States competitor, but the approval was easier or quicker in Europe. Can you translate that to US?
Sorry, I think as previously stated, if you can show why those lots are also comparable, highly similar, you can meet that to the US license reference product. And if you can bridge to the EU product, you have to show us why that data that's relevant to EU reference product, why that's relevant to us, why that's referenced to the US license product and its prescribing information. Does that make sense? Help?
Okay, Ray, another online question, please.
What happens if an approved innovator biologic product is taken off of the market by the seller? Would a 351K still be a possible pathway for approval for that product? And if so, what would be the reference product?
So excited to talk and felt left out, and now I don't want to talk. So we're still working through various aspects, various questions that we haven't quite received yet. There's a lot of regulatory implications that we need to discover and examine before we can provide any outward-facing guidance to answer that question. Something that may come into play would also be, was it withdrawn from the market for reasons of safety, purity, and potency? Was there an issue with that product? So it's still under discussion, and as soon as we have an answer, then we would release some guidance.
Okay, thank you. And it looks like I'm getting the shake off from the catcher back there. There's no more online questions. It doesn't look like there's more in-room questions. So please help me thank our panel for sharing their wisdom with us.